First of all, I would like to thank you for giving me the opportunity to make a presentation at such a valuable congress. As you know, the pandemic has caused radical change and destruction both in our world and in every region we live in. In this context, study and efforts have made on ways to get rid of this situation. Is to survive with the least damage and from different angles and aspects by taking both vaccines and or drugs and different health measures. Sorry. Science Pincer types ligands L1 and L2 and their metal complex, copper metal complex, were synthesized and characterized by using different techniques. The products of reaction with the related ligands were new tridentate Pincer complex. The molecular structure of the copper complex were estimated by X-ray and molecular conductivity measurements, in which they exhibited a trigonal pyramidal geometry with 5 coordinates around their metal centers. Furthermore, the current compounds as a potential drugs candidates agents SARS-CoV-2 were investigated via molecular docking simulation. Why we select the copper metal to generate complexes with the CNC Pincer ligands? Because copper is the economical agent which is a cheap. Copper is involved in a function of a critical immune cell, and the lack of copper is associated with the increase in infection rates. Generally, metal complexes have a wide range of structural change and diverse stoichiometries due to their multiple specific ligand combination. They are drugs with strong adaptability. The use of antiviral drugs in combination with the metal drugs can enhance efficiency and reduce side effects. For example, quinoline-based copper-2 complex are designed and synthesized as a potential drugs against COVID-19. You can see that the genome-wide structure and function modeling of SARS-CoV-2. We can focus on the non-structural proteins and structural proteins. And to date, a variety of antiviral drugs, anti-inflammatory drugs, antibodies, corticosteroids, various herbal marine agents, and convalescent plasma have been recommended toward the treatment of COVID-19, whereas effective drugs are still under screening. Mounting evidence indicates that the development of effective vaccines is the most promising route to stop the pandemic. Until the end of May 2021, the 6-month vaccinating worldwide has resulted in a strongly curbed spreading of SARS-CoV-2. However, due to the unevenly distributed vaccine research and the long-term side effects caused by COVID-19, there is still an urgent need for drugs to combat this disease in an effective, safe, stable, and reliable way. As a potential metals and metal compounds have attracted widespread attention due to their unique pharmacological advantage in efficiency, safety, and stability. Some metal elements such as selenium, iron, and copper are known to interrupt the infection interaction between the virus, the host, preventing the virus from entering the host and or elevating the symptoms. In addition, metal compounds have various chemical structure and exhibit wide range bioactivities. Metal drugs are known to play an important role in a direct antiviral or inhibiting critical enzyme in a process of virus replication for the SARS-CoV-2 infection. Metal drugs can also be used as adjuvant drugs to enhance the efficiency of antiviral drugs or used as a vaccine adjuvants. It has been illustrated that many metal nanoparticles and metal complex exhibit excellent anti-inflammatory and antiviral activities. To date, many antiviral small molecule drugs have already been proved to be in the treatments of severe COVID-19 patients. For instance, or for example, dexamethasone was found to reduce deaths from SARS-CoV-2, and remdesivir was demonstrated to shorten the length of the hospital stay of the COVID-19 patients. In this research, molecular docking was applied to describe the combined L1, L2, and their complex in the SARS-CoV-2 target model. The related models were primarily determined in accordance with structure properties of the virus, as well as based on biological mechanism and function that can be benefited to reduce, hinder, or therapy of the virus. You can see that the binding affinity of the Pincer ligands and their copper-2 complex against non-structural proteins in the number of the 15, and also the red color, you can see that the structure protein, such as spike protein, is analyzed in this study. As the end of the calculation, the binding affinity result is summarized in this table. End of the docking process, the compound L1 display the best affinity minus 10.40 kilocalorie per mole with non-structural protein 14. Afterwards, the same compound have respectively second minus 9.60, and the third one is the minus 8 kilocalorie per mole with the non-structural protein 3 and non-structural protein 16. You can see that the 3D views of this best interaction. As seen, figures complex of the compound L1 with the non-structural protein 14, C-terminal guanine and 7-methyltransferase is described with the following interaction. Two types of carbon and conventional hydrogen bonds with the alanine and asparagine and glycine, and also beside hydrogen interaction is available halogen bonds. 11 halogen bonds is shown that these parts, and again these parts again is you can see that with the trifluoromethyl parts, you can see that in the both sides. In the meantime, hydrophobic interaction is available, which including the PP stacked interaction, alkyl interaction, and p-alkyl interaction is available. In addition, one p-sulfur interaction with the cysteine residue of these targets. The second best interaction is the, you can see that with the non-structured protein 3, there are 4 hydrogen bonds with the residues, you can see that. And also is including the, this slide is summarized the hydrogen bonds hydrophobic and solvent accessible surface is summarized in this slide. There are again hydrogen bond, halogen interaction, and hydrophobic interaction without P sulfur interaction. The third one is the lastly, the current compound give the 3 hydrogen bonds with the tyrosine and this residue. 1 halogen bond is available and 1 P sulfur interaction is available, not cysteine with methyl residue. And also hydrophobic interaction is available. How do you say that? Because the hydrophobic interaction surface and also hydrogen bond interaction surface and the solvent accessible surface is shown which types of interaction is supported in this slide. And besides the non-structured proteins of the SARS-CoV-2, spike protein, which is the structural protein, is of importance. The surface spike glycoprotein is consisting of 2 heterodimers, S1 and S2. The receptor binding domain, or the other word is the RBD, is located on the head of the S1 and binds the cellular receptor angiotensin converting enzyme 2. In other words, is the ACA2, initiating the membrane fusion of the virus and host cell. At this point, 8 mutation, you can see that in the RBD and human ACE2 interaction region were used to investigate the interaction mechanism of the reported compounds towards spike protein, RBD, as a target. Based on this approach, the current compound exhibits similar behavior against the native and mutated form of the spike protein in the molecular docking study. These are expressed by a red rectangular frame, especially L1 and L1 copper complex. How do you say that? You can see that this part, as a result of the calculation, spike protein, one of the structural proteins of a SARS-CoV-2, was docked with the respective compounds to evaluate against the current variants of SARS-CoV-2. Based on this approach, especially L1 copper 2 or copper metal complex exhibited the best behavior against the native and mutated form of the spike protein in the molecular docking studies. And you can see that this slide, the binding affinity of the spike protein with the mutated RBD was slightly decreased towards the binding affinity of the copper complex, the native spike protein, from minus 6.97 to minus 6.87 kilocalorie per mole. In the second part,
1 to 2 minutes. 1 to 2 minutes.
Okay. In the second order, the compound L1 exhibit the second best interaction with the same native and mutated target. The same orientation and interaction of the target in the is demonstrated, and the binding affinity is smaller than the copper complex. This shows that the activity of the related complex maintains the same in the both conditions, as you can see that from the 3D interaction image. And you can see that the results, based on the results, L1 and copper complex were exhibited by energy. We can summarize this part. And also, these findings show that the ligand L1 and copper complex have a high potential inhibition against in non-structured proteins, and even also the native and mutated form of spike protein, which is one of the structured proteins of the SARS-CoV-2. Based on the various findings of the related complex, the development of these complex may open a new area of the metal drugs, although there is still a lack of data and clinical trials to rate of efficiency and safety of metal-based drugs in the curing COVID-19. Further research and evaluation remain necessary. Thank you for your interest.
Thank you very much. We have one question from Dimitri Shulga. How was copper treated during docking? Did you derive the parameters or use some existing ones?
Yes, we can interact with the how was copper to treat during the docking. We can dock with the docking software to such as AutoDock, you know that. Not derivative. We can use the copper ion and interact with the ligands, CNC, two types of CNC ligands, and also copper complex. And you can see that the in silico analyzing against the COVID-19.
Okay, you have performed docking studies, and how can you ascertain the stability of interaction between the ligand and target? Did you try to perform molecular dynamics simulation?
We can organize in the future time, not this part.
Okay.
Okay.